Hello, my name is Roberto Martinez and I'm representing Phoenix College. My research is on the colonization and time-dependent biodegradation of microplastics by Pseudomonas aeruginosa. Plastic pollution has become an increased concern. Recent studies have reported that drinking water around the world contains microplastics. These are small pieces of plastics less than 5 millimeters in diameter. Currently, microplastics are not being removed by wastewater treatment plants. One proposed solution is the use of microbes to biodegrade microplastics. Pseudomonas species have been found to grow on many plastics. The purpose of this project was to determine if Pseudomonas aeruginosa is capable of colonizing and biodegrading polyvinyl chloride, low-density polyethylene, and polypropylene. The findings of this study have implications for the bioremediation of microplastic contamination and environmental water and soil samples. 140 million tons of plastic are produced every year. Figure 1 above shows the percentage of tap water samples contaminated with microplastics by region. As shown, the U.S. has 94.4%. The average amount of synthetic debris consumed annually from tap water, beer, and sea salt is over 5,800 particles, with 88% coming from tap water. Pseudomonas species are gram-negative biofilm-forming bacteria that can colonize plastics. And some Pseudomonas species have been reported to grow on polyvinyl chloride, or PVC. For this study, we have chosen to utilize the three most common household plastic products. Recycling code number 3, PVC, or polyvinyl chloride, is used in plumbing pipes, blister packaging for non-food items. Number 4, LDPE, or low-density polyethylene, is found in food bags, squeezable bottles, films, and disposable cups. Number 5, PP, or polypropylene, is used in microwavable containers, yogurt cups, disposable plates, and drinking cups. We hypothesize that Pseudomonas aeruginosa can colonize and aid in the biodegradation of microplastics, PVC, LDPE, and PP. To produce our microplastics, we used punch pliers soaked in 100% isopropanol to remove microbial contamination. You can see our produced disc in this second image here. Pseudomonas Isolation Agar and Pseudomonas Isolation Broth were used to isolate Pseudomonas. Alcohol-treated microplastics 3, 4, and 5 were co-incubated with pure cultures of Pseudomonas aeruginosa and PIB and 0.1% peptone broth for 7 to 125 days at 25 to 37 degrees Celsius. The co-incubated discs were then transferred to PIA plates and observed for colonization. Scanning Electron Microscopy was used to confirm colonization of our microplastics. An infrared spectroscopy FTIR was used to analyze microplastic samples for biodegradation. Figure 2 shows SEM analysis of microplastic colonization. As we can see here, the top row is our control plastic and the bottom is our co-incubated microplastics with Pseudomonas aeruginosa for 7 days. We can see that there's a biofilm formed at the bottom row, which, when dried, form these cracks. We also see bacilli structures, which confirm the presence of our Pseudomonas aeruginosa. Figure 3 also confirms colonization of our microplastics. The top row is our control, and bottom row shows our PVC, LDPE, and PP colonized with Pseudomonas aeruginosa. 4, 5, and 6 show our FTIR spectral overlay of are plastics PVC, LDPE, and PP. We can clearly see changes from the first timeline of 28 days to our final timeline of 125. Many of the characteristic peaks lower in intensity over time, while we get newer peaks increasing in intensity. These changes compared to our control confirm that the colonization of our Pseudomonas aeruginosa is having an effect on the surface of the plastic. Figure 7 is an SEM analysis of microplastic biodegradation of plastic 3 PVC co-incubated with Pseudomonas aeruginosa at 28 days. We can see a significant difference in pore size. Due to the porosity of PVC, Pseudomonas aeruginosa can anchor itself into these holes and better colonize the PVC surface. We believe this is the reason for our increase in porosity shown on the right side. Pseudomonas aeruginosa was tested for the ability to colonize plastic types 3, 4, and 5. Colonization of plastics 3, 4, and 5 was observed by growth on PIA and observed by SEM. Preliminary FTIR data shows biodegradation of plastics by Pseudomonas aeruginosa. For future studies, we hope to use nutrient deprivation to explore metabolic pathways involved in Pseudomonas-mediated biodegradation of plastics. We can also expand our study to include other Pseudomonas species like Pseudomonas fluorescens and Pseudomonas alkaligenes. And we will expand our study to examine microplastic degradation for 150 to 180 days. Research was supported by the Western Alliance to Expand Student Opportunities, Lewis Stokes Alliance for Minority Participation. Thank you.